hate how month doesn't start with the month number that is. If it's October, should it be?
All right. Hello, everybody.
Yeah, the month. Yeah, whenever.
So just a quick little update. I have some exams still, so if you didn't get yours last time, come see me after class. So I've got your graded exams. I haven't put the grades into Moodle yet, but I'll plan to do that and update homework grades and all that the next, hopefully by next Tuesday will be my aim. So you'll have all that in Moodle as well as your own records. Okay, so today we're going to talk about membrane filtration. Last time we had a lecture was on granular filtration. We kind of wrapped up the quantitation aspects of granular filtration, talking about how often are they running, how do we account for the downtime, all of that. So today, whereas last time we talked the granular filtration, we had these like packed beds basically with a bunch of granular media, put water on top. Well, now we're going to talk about primarily size exclusion. If we get low enough, then there's some other principles in play aside from just size exclusion. But by size exclusion, I mean we have some particle, and we're going to pass it through a membrane, which is essentially a flat sheet. It's porous to some stuff. So for the water, or in the case of like a screen porch and mosquitoes, it's porous to the air, not porous to the mosquitoes, right? That's the hope. And so as you pass the water with the particle through, the particle should then get stuck on that surface. So it's blocked from passing through. So kind of had the same figure when we were talking about when we introduced granular filtration. So this is the difference. Now we are really dedicated to size exclusion as a separation process. All right, so what is a membrane and what is all this about? So kind of a dictionary definition of a membrane would be a thin, soft, pliable sheet or layer, especially of plant or animal origin. So some membrane, that part of the definition is just kind of the physical aspects of it. It's thin, flat, pliable. Another definition could be a piece of parchment forming part of a roll. So, I could take these sheets of paper here, well, so I don't show the class somebody's exam. I'll grab some other paper, and you can treat it then as this could be a membrane, right? It's thin, it's pliable, it's flexible, it's a sheet, right? It's a layer. So we can do different things with that, and in the terms of most membranes, one of their functions is to allow some stuff to pass through, right? So a cell membrane keeps a lot of cellular components inside, but would also allow certain nutrients or stuff to pass through that cellular membrane so that the cell can have water, sugar, whatever it needs to do. It can take stuff in, it can send stuff out, has some control over what's going in and out. So I would add to this, a membrane typically also has control over some stuff passing, but not everything. So in terms of utility, what we use them for, particle removal, just like we do with granular filtration. We don't particularly need a very fancy membrane for that, but we can also get a smaller pore size membrane and then start excluding really small suspended colloids, remove viruses. That gets us down to basically ultrafiltration, what we might call, whereas if we're just doing larger particles, we might call that microfiltration. Then we could get down to nanofiltration, and it's a little bit of a spectrum here, we'll see in a minute, where we could start removing some large chemicals. So if we've got organic matter that's just in the water by nature, something like you would expect if you were to visit a river and it's stained brown or something, those molecules that are causing that color are individual molecules, but a lot of times they're rather large. So you could potentially remove those with nanofiltration. Or you could go even further and get down to what we would call reverse osmosis, where you can reject ionic species like salts, right? So you could reject actual salt with the membrane if you give it enough force. You don't get a whole lot out, so it's very expensive in terms of energy, but you can absolutely desalinate water with reverse osmosis. So if you're ever considering people trying to scare you about water scarcity, well, assuming there is access to some sort of water, which is not necessarily the case for a desert, but assuming there is water that you can access, there are ways to clean it. It just is going to cost, right? So water scarcity is more of a function of infrastructure and energy costs than of actual water being the issue, unless, of course, there's desert situations. Okay, so with relevance to water treatment, membranes are actually as simple as it sounds. You don't let something through by having this screen type of a situation. Membranes haven't been used for all that often. It's only first used about 60 years ago in water treatment, and really more commonly popularized about 30 or so years ago. So it's definitely pretty well developed, but it's not as old of a technology as, let's say, chlorination. One of the things I think we talked about a little bit, we will definitely look at a graph in a couple minutes, is that the pore size for these membranes, so you can think of the pore size as how big are the grids, right? You can look at the back of the chairs you're sitting in, and they have a screen mesh sort of thing, and mosquitoes could definitely fit through that, right? That wouldn't work for a screen porch. If you're thinking about it in terms of the pore size of some membrane, lots of different ways to create a membrane, but you'll have some sort of typical pore size that is your limitation. And the smaller the pore size, the higher the pressure you need to push water through, right? So the pore size is inversely proportional to the pressure required to operate it. Likewise, the amount of water you get permeating through it, so the permeability, is also inversely proportional to the pore size. So we can do reverse osmosis, but a lot of pressure for little water is the thing there. Okay, a few operational terms we need to make sure that we have clear so that we can understand what we're talking about. We'll talk about these all the time for membranes. First one would be the permeate. That's the water that permeates through the membrane, right? It's the produced, cleaned, permeate water. So if we had a membrane and water passed through, then that would be the permeate. Then we have the feed, which is pretty obvious. It's the water that we're feeding to the membrane. And then sometimes we have a separate category of water that did not make it through the membrane. So either we pushed some water through but kept some back, or we have what we would call a cross flow, which we'll talk about in a moment, where some of the water is intentionally not going through the membrane, so it can carry away all the dirt and particles, so that that's not all just stacking up on the membrane itself. So a lot of times we'll have three flows: the feed going to the membrane, permeate going through it, and a rejected stream, which we would call the concentrate or the retentate. And that is going to be a concentrated dirty water, right? So the mass balance here is we have some amount of mass of particles, contaminants, whatever we're dealing with, and some amount of water, neither of which are going to change in the system. It's just we're changing the directions. It's a lot like when we mix two streams with different concentrations of something. They come together and make a final stream with the combined mass, except we're doing it in reverse, right? Since we have a separation process, we are taking one stream and splitting it into two different. This is like the one time where we can split a flow rate and have different concentrations either side. Normally, if you take a pipe, you split it two different ways, the concentration won't change. It's just two different directions, right? Same water. With a membrane, we can do that, but then control how much is going one way versus the other. And so this would be that. That's what I'm referring to when I say concentrate or that retentate. That's the stuff that was retained and that did not permeate through. And as I say here, this is extra dirty. If the feed is dirty water, then the concentrate is extra dirty. Now, of course, if you think about it, you've heard the name concentrate before. If you've ever bought orange juice or looked at a label, it will say from concentrate or not from concentrate. So essentially, what that's saying is if orange juice was being produced and then they decided to ship it across the country, they realize that it's a lot cheaper if you don't have a whole lot of water in it. So what they'll do is they'll pass it across a membrane, concentrate all the good stuff that we want to drink, and get rid of this clean water. That's not what they need it for. You might lose a little flavor or something, so I'm sure they try to control it, maybe use the right kind of membrane to retain all that. But then you squeeze basically all this water out of it, concentrate it up, ship it across the country, and then reconstitute it with pure water. And then you've got presumably the same stuff you started with. It just costs a lot less to ship, right? So that's pretty much the whole reason you do that concentrate or from concentrate, or maybe not if you think that maybe you're losing some flavors in the permeate, which is possible. It would depend on their membrane selection and all that. Okay, so to put this in more technical schematic terms, typically what we'll do is we'll draw some sort of a diagram and then add a line through it, some sort of diagonal, to illustrate there's a membrane that the water has to cross there. Okay, so it's not a very technical diagram, and I have 4 arrows here just to show you that it's kind of arbitrary if I go from the bottom right to the top, bottom left to the top right, or the top left to the bottom right. It doesn't really matter. It's just we will only need 3 of these arrows, right? So in this case, this is basically the same as this arrow, so I could just mark that off, right? So in this case, the way I drew it, this is the permeate, and this is also a permeate, right? That's what I'm saying. It doesn't matter. I could cross one out. I could cross this one out if I felt like it. The point being, water past that little line that I drew, right? So if I'm drawing a schematic like this, all I'm showing you is, all you should care about is which way is the water going, right? The water is coming in. It's kind of obvious that'll be the feed. Whichever direction finds the membrane and bounces away, does not pass through that line that I drew, that's going to be our retentate or concentrate. Okay, so then whatever does make it through in our little box diagram here is going to be our permeate. Does that make sense? Okay. The other way to operate is the simpler version, where we have a dead end. We have a filter here at the bottom, typically, or at some point. If that's our filter, water is coming to it. We're feeding it water. And maybe we stop filtration at some point and we can retain some concentrate in that little space, but it's not, we don't have a continuous flow of concentrate. We'd have to intentionally stop before filtering everything. So this is more like a lab scale thing if you've ever dealt with a little syringe filters. It's pretty common in environmental labs, at least, where you have a little syringe, you get like 50 milliliters or 20 milliliters or something, and you can attach a filter on the end, and then you push it through with your thumb. And if you clog it too much, then you're like really, really trying to push. It's a really good workout for your thumb. And then your filter breaks and sprays everywhere, and you're like, oh shoot, I screwed that up. Right, so there's a pressure dynamic and fouling there. We'll talk about all that. So this would be dead end, and as that little story sort of thing illustrates, it's not great for continuous operation. Right, this would be sort of like a batch reactor in the sense where you do it for a little bit and then you're done, and that's that. Right, you'd have to then probably change the filter, do it again. Like the syringe filters are disposable basically, so you use it once and you toss it out typically. Whereas the cross flow or continuous, which is this guy, that's designed for continuous flow, continuous operation, all of that. So this one's the cross flow, distinct from this dead end filtration. Okay. And that would be the membrane. All right, so cross flow filtration then is, we could illustrate it here where we have a membrane. You see these pores being the spaces where water can go through. And you see we've got this feed water, it's dirty, meets the membrane. Membrane's hopefully rejecting all these particles. And then this retentate is extra dirty with those particles, carrying them away from the membrane so it doesn't get fouled up as badly as it would have. Because if you can imagine if we close this off, so there is no retentate, then we're in dead end, and all of these particles will just accumulate on the surface here until at some point we just can't push any more water through because the particles themselves are acting kind of like a filter. Okay, so that was kind of cross flow. I think maybe I inverted these by accident. Dead end would be the other one. I talked about it. This is kind of what it looks like. A syringe filter with some sort of liquid in it, pushing the water through. You got some sort of a membrane there. This is kind of what I was drawing. If you leave some water in there, let's say you start here, and then you end here, whatever's remaining and whatever you keep here could be the concentrate, right? That would be this if you left it remaining after filtering some out. Okay, we'll look at a little problem that describes, kind of gives us an example of that in a little bit. Okay, and like I said earlier, doesn't matter which way you draw this so long as you're watching where it goes through. In this case, the way I drew it, this would be concentrate, retentate, and the permeate would be on the other side. And you could just, cross that one out if you wanted. All right. So another example of how a dead end filtration might look in practice. You'll see these in labs as well. If you want something that's slightly more sophisticated than using your thumb to pressurize a system, one thing you can do is you put some water in one of these flasks. You have a membrane support, and you put a little membrane on top of that support, and then you pull water through, partly with the gravity, but also by applying a vacuum. So if this is our membrane here, then we would apply a vacuum to pull the water through, and we'd get a permeate here coming out, collected in some beaker or something, and then you would call this your feed. And of course, if you stop filtration while you still have some of that feed water, that becomes then your concentrate, right? Because you've concentrated it by rejecting some of the particles. They're stuck up there, and like we talked about. Something interesting to note here is if we do use a vacuum for filtration, we have an inherent pressure limit. So if we're using a vacuum, then the maximum pressure we can apply is 1 atmosphere if we're doing this in a standard 1 atmosphere environment. We could go to Jupiter and have a lot higher of a pressure if we pressurize the vacuum down to essentially 0. But on Earth, we've got one atmosphere worth of pressure typically to work with, and that would be our pressure that we would have here. So the delta P for the pressure for vacuums is max of 1 atmosphere, right? So we can certainly create pressure with a pump that's much higher than 1 atmosphere, but if we're going to do something like a vacuum filtration setup, that would be a limitation potentially. So in this case, the water should hopefully all go down and not get pulled by the vacuum. Of course, that's somewhat of a risk there, but generally you'd operate it so that you catch pretty much all the permeate, because you don't want the liquid going into your vacuum. Okay, so here's a lab scale demonstration thing of a cross flow system. Found this image online, and essentially what we see here is a membrane setup. Let's say what we would call a tubular membrane, where we have a tube, hollow tube with some wall thickness, and in this case it appears that we're feeding from here. So we've got this feed, this dirty water here. It's being pulled by this blue pipe into this vacuum, or not vacuum, sorry, this pump. And then from the pump, it's being pressurized into here. And so we're applying a pressure, and inside this little chamber here is kind of the membrane. So we could potentially think of it, you know, if we were to draw the box diagram, it's not going to work very well. But we could think about it as the actual process is happening in here, where we have, I guess it could make a little bit of sense this way, where we have the feed coming in, the permeate is making it through, but actually the retentate, I would have to draw it something like this, I guess. So the permeate makes it through that line, and the retentate continues on its way, because then the retentate we can see is coming out here, or the concentrate, and that's dirtier water. It's a little hard to see how dirty it is, but you know, if it filled it up further, I'm sure we'd see it's dirtier, and we can see this permeate is reasonably clean comparatively. Yeah.
So is there some hollow section inside of this filter? It's just very pressurized that pushes it out?
Yeah, so I was meaning to come right back to that. So the way the filter is working on the inside here is going to be the hollow tube is itself a membrane where we have, I think the pressure's coming, the water's coming into the inside of it, and then some of it's pushing out, and this is the membrane surface. So the water is just continuously pushing from the inside of this tube to the outside and leaving the tube. Some of it, of course, passes all the way through the tube. And what we have over here is a pressure valve to pressurize it. It's going to prevent most of the water from coming through to the concentrate. So we have a pressure valve here. And then the other thing we have is a kind of a clear plastic. It's a little hard to see. Clear plastic lining outside of the membrane tube that's collecting all the permeate water. And then it doesn't really matter where it comes out of this tube. So it's the water is coming in and it's working its way out of our membrane, some of it at least. It doesn't really matter where it comes out because it's all going to be collected into this sheath and then funneled down into our outlet right here. Does that make sense? Does that answer the question?
Yeah.
Okay, so it kind of gets interesting when we take a look at these membrane systems because there's a lot you can do with the geometry, right? So in this case, and what we'll see in a little bit, is you could take what we would call a flat sheet membrane and then curl it around as long as you have a way to support it. Maybe have a little support tube, and then you can do just this type of filtration right there, right? You pressurize on the inside, force it on the outside. So there's a lot of kind of interesting geometric manipulations that you can do with membranes in order to apply it in a particular way. But the same principle applies in terms of you have water fed to the membrane, some water permeates, some water is rejected. It's just the plumbing of it all can look different, right? So that little box that I drew at first, and we went over a million times already, it's like that little box is going to apply because we have a plane that separates some water from some other water, right? That's our separation process happens right there. Okay, so we talked a little bit about it already, but the pore size of our membrane, the way we choose that is really going to be based on the contaminant that we have to remove. So typically when we design a membrane system, we'll think about, okay, well, what are we trying to remove? That contaminant size, then we need to make sure that we have pores that are no larger than that contaminant, probably that are smaller, in fact. And so that then we can know, okay, well, that means we need this type of a pore size, and that's going to tell us, oh, okay, we need this much pressure in order to achieve the flux that we need. So there's there's a couple things, right? We might need to treat a certain amount of water, which means we need some ultimate flow rate, and we can get a flow rate based on some sort of surface area of our membranes times a flux, so the flux being the speed at which water is permeating through them, just taking us back to, okay, how much pressure do we need to provide to find this, to get that flux? So all of that put together kind of tells us, if we need just very simple filtration, like basically the equivalent of granular filtration, probably don't need a whole lot of pressure. We can push water pretty quickly through because they're large pores, and we can design it that way, versus if we need a really high selectivity, so we don't let much through, we're going to need a lot of pressure. And then we're going to need a whole lot of surface area to apply that pressure to if we need to produce a lot of water. Okay, so all those things are sort of tied together, and that's kind of the design process is, okay, well, what do we need to remove? That means we need this kind of membrane. That means we're going to have to apply this kind of pressure, and we know then we'll get this kind of flux. Therefore, we need this many membranes. So all of that kind of goes together there. There's actually a couple ways you can potentially operate membranes. You could say you could operate in constant flux or constant pressure. So you could make sure that you're always going to have the same amount of flux. That's kind of the typical for practice in terms of, I want to make sure that I'm always giving my municipality the same amount of water that they need, right? You could also do constant pressure. That's sometimes useful in laboratory settings to study membranes, but not usually applicable to a real system. Maybe you would consider a case if you're doing like a gravity-fed operation and you're using a set height of water, then you're going to end up with a constant pressure, and you're going to try to achieve with that a relatively constant flux, or maybe a storage tank to even it out or something. So there might be cases where you'd have constant pressure, but normally we try to operate with constant flux. Okay, both aspects are going to be important in terms of knowing how much water we get out of it. Okay, like we talked about with the granular filtration, even if it's a membrane filter, whatever it is, we're going to have some flux through a surface area. So that same concept is coming back. Flux is a flow rate through an area. So that you could look at as cubic meters per second per square meters, or meters per second. It's all of that's in there. And so the same ways of thinking about the granular filters that we considered apply here, but we're going to go a little further in terms of defining specifically how does that interface with the membrane characteristics. All right, so we looked at one chart a little bit like this when we were talking about filtration generally. Here's one that's a little more specific to membrane filters. Well, we do see the conventional granular filters up here. So what we're looking at is essentially the effective pore size on the top. So pore size is kind of our x-axis, so to speak. So 1 micrometer here versus up to 100 micrometers or 0.1 millimeters starts getting to the point where you can tangibly think about that. And that's about the size of sand grains up here at the top. See that? And then you can kind of go down the list and say, okay, well, the microfiltration membranes can be used for the same things a granular filter can, and for a little smaller. So you can start getting into some bacteria removal, maybe some virus removals, but that's kind of getting into the territory of ultrafiltration. You can remove pretty much, with a decent microfiltration membrane, if you're above 1 micrometer, maybe you're not removing all bacteria, but you can certainly call it a microfiltration and do 0.22 micrometers, and that should remove just about all of the bacteria, and so on. Right? So you have ultrafiltration, you've got nanofiltration, all the way down to reverse osmosis, where you're really getting down to less than nanometers, so the angstrom level pore size. So water can get through, but not much else can. This water is very small. There's some other issues. Actually, the space station has some issues with very, very small levels of methanol and ethanol being formed just from biological processes and whatnot occur in their water system, and their membrane filtration systems can't remove it because it's so close to the same size as water that even though they're using a reverse osmosis, it's just passing right through. So there's some interesting investigations on how to prevent the accumulation of ethanol in their drinking water. Would be a kind of an odd thing to think about, but there are some processes that you might get a little formation over time. So if you look at the pressure requirement versus the flux you're getting, I mentioned it, but it's kind of interesting to look at. So if it takes 100 kilopascals, and by the way, it's 9.8 kilopascals is essentially 1 meter of water at the Earth's surface. That's the amount of pressure you get having 1 meter of water standing on top of you, basically. So that means that's about 10 meters of water here to get to push something between microfiltration and ultrafiltration. So that would be 10 meters of water here. And so there's actually a neat system called the sky hydrant. I had a chance to take a look at and play with a little bit, where someone designed, I think it was GE, designed a large microfiltration system, or I guess ultrafiltration system. Not large. It was like about this tall, so a unit like that. And essentially, it was designed to be hooked up to a storage tank that was elevated about 10 meters, 35 feet or so, right? So you could imagine building a structure or having a tank on a hill and then feeding that pipe down to this filtration unit. And the idea here was that would be a reasonably sustainable way to get water treatment for perhaps like disaster relief or some kind of emergency situation like that. And you could toggle the valves to do backwashing, and it could just simply use that gravity-fed pressure, which gravity is going to require energy, right? You have to have a way to get the water up there in the first place. But if you have that set up, then you can use that as a continuous source of pressure for the membrane itself, which is removes a lot of technical challenges. So that's pretty neat, and that you see that can remove most viruses, perhaps even all viruses, depending on how exactly the system was set up. And that would be a huge improvement to water quality. Now, maybe there's still some concerns. You could still have maybe heavy metals that you're worried about in particular areas. Definitely not going to do anything for that. If you did want a membrane to do that, you'll be looking at thousands of kilopascals required, and your hydraulic flux went from, close to 100 liters per square meter of your membrane per hour, down to between 1 and 10, once you're down to like kind of the nanofiltration sort of stuff. So if you want the same flow rate of water coming out, that means you're like multiplying the amount of membrane surface area you need by like 10. Okay, so the smaller the pore size, to meet the same amount of flow, you're just going to need a lot more membrane, a lot more surface area. Okay, so how do we make membranes? What are they made of? Like, what are these things actually? Well, the definition said they're often plant or animal derived, or that's that typical membranes you'd see, cell membranes and stuff. Well, we actually do get a lot of plant material in water treatment membranes. So polymeric or polymer membranes are one of the most common. The first one here is cellulose, kind of shows that because essentially cellulose is coming from plant material, right? If we process it, we can then use that. It is a natural polymer. It's relatively robust. You think of trees are made up of stuff that's supposedly should be sturdy and not going to dissolve as water is passing through them, right? So it should be we should be able to use cellulose pretty well. That is the case. It works out as a pretty good material for us. We have other ones. We can use PVC, polymethyl methacrylate, so acrylic stuff, all sorts of other ones. There are many polymers that are used, variety of different reasons. Maybe you want a surface charge that's going to prevent fouling somewhat. You can prevent particular types of organic matter from passing through it, or prevent particular types of organic matter from sticking on it and fouling it. Trouble is, we have a little bit of a limitation on cleaning. So the good things are they're typically pretty flexible, easy to handle, easy to create in whatever design, shape, whatever we want. But they have typically some temperature limitations. If we get the water too hot, if we're cleaning it with hot water, then we find a situation where we're melting them, right? If we try to apply too much pressure, we're going to collapse them. So we have to be careful with how we design them to deal with pressure. There are some good membranes that can handle high pressure, but there's a lot of care that goes into making sure that they don't just go splat, right? And you can't bleach them too harshly, right? Because some polymers are going to react with the chlorine itself or whatever chemical cleaning agents you're using. Potential to oxidize the membranes or destroy them, dissolve them, that type of thing. An alternate would be ceramic. This is a little bit newer. In one sense, it's very old, right? Ceramic technology is quite old. You put some clay in the oven and bake it, right? But with kind of more modern design capabilities, we're able to fabricate with pretty good control membranes out of ceramic materials, which gives us a lot of answers to those problems, right? On the flip side, it's brittle, so it's going to be easy to break. You have to carefully control the way you're handling it. But once you do have it set up, you can typically handle quite a bit of pressure. This is sort of a robust inorganic framework, basically mineral, right? So higher pressure, chemically resistant, heat resistant. And when I was a grad student, I had a lab mate, a master's student, who was working on ceramic membranes for treating the waste stream of a contaminant capture for a coal-fired power plant. So flue gas, they basically use water to strip some of the contaminants out. Then you'd have this dirty water that's also very hot, so like 60 degrees Celsius or something. So pretty hot water, and it's you would have to normally cool it before treating it with a membrane. But there's actually some advantage if you can use it as hot water, because if you remember, we'll talk a little more about this, but our viscosity is going to be related to how easy it is to push water through some porous material, right? We've seen viscosity important in several things by now. And so with high temperatures, the viscosity is lower. It's less viscous, meaning that's actually probably an advantage for membrane filtration, right? It slips through the pores easier. So if you could keep it hotter, you don't have to pay as much energy cost for the membrane. You don't have to cool it first. So that's kind of another process advantage. So it's kind of an interesting project. I don't know if they are using it or not, but it seems like if they figured it out correctly, then they probably would. Okay, so what about the physical form? We took a look at that lab demonstration one. Here's a kind of an interesting one that's actually quite common. If we use a flat sheet membrane, like a piece of paper, and if you think about it, we take this flat sheet and we want to pass water through it, we have to apply water to that entire sheet, collect it on the other side. And so if we keep it as a flat sheet, then the best we can do is put like two walls around it with kind of white water pipes to either side. And then we've got this somewhat thick unit that's taking up a fair bit of space, right? And then you just have, if you needed 200 square meters of membrane area, then you basically need 200 square meters of these devices, right? That's not very efficient for space. But you could wrap it In fact, you could wrap it with several membranes. If we take a look at what I'm doing here, we can take it and wrap it with multiple sheets. So long as we have the correct plumbing in between to make sure that once water passes through, it's guided along through a kind of a freeway sort of thing inside, and that's what you see in this picture up here. There are channels inside these layers, right? So there's, in the layers, once they pass through a membrane, then they'll be allowed to spin their way into the middle to be collected. Otherwise, water is just going to carry on through. So in this case, we have a flow coming in, the feed. Can't see my marker very well. The feed coming in to the sides here. So it's as if I'm putting water into the inner spaces between the membranes. Once it passes through, it can't get back because of the way we plumbed it, and it can only go through until it is collected inside this tube, and then we have the permeate coming out here. If this tube goes all the way through, we could also collect the permeate here, as we see. Whereas the retentate, kept separate from that permeate, still is going to be coming out from between the sheets of membrane, just out the other side. So it's all kind of about how you plumb it inside, but once you do it, it works, right? So you just have to keep the feed separated from the permeate, and you can kind of see how it works here. It's a little bit not intuitive at first, perhaps, but it certainly does work. You know, we certainly still have the ability to essentially cause our system to act like this, right? We have water coming in, water going through, and water not going through, right? So that's the whole system. It's just wrapped up in this, what we'd call a spiral bound membrane cartridge, or spiral wound. Okay, another form is this tubular membrane. We saw one example. We had one tube housed in that little clear plastic container collecting the permeate. You could put multiple tubes in a container like that. Doesn't have to be clear plastic, but it was kind of a good way to see it, if you can see it from that picture. And you could operate this in either way, right? You could take water coming into the tubes, and if they make it out of the tubes, that's permeation, pass through the filter, and then you collect it. Or you could reverse that and use the outside of the tube as a membrane and do outside to in type filtration, where you just reverse, have the feed here, permeate coming out through them, and then the retentate would be whatever water did not make it into the tubes. In this case, we have water coming into the tubes, and whatever water does not make it through the tubes, like through the spaces in between, will just come out the tubes on the other end. Again, we'll have a pressurization device there to make it so it's not just easier to flow out, right? So we control that pressure, control the flow heading out. But for this segment of the membrane, we can understand it in this manner. Does that make sense? Okay, so a lot of this is about how you pot, is what it's called if you've ever done like electrical work or some piping work, membranes like this. You call it potting, where you seal off any outlets, so that in the case of electrical work, maybe you're sending electrical cords through the wall of your house or something, and you want to make sure bugs don't come in. Then you add some foam or some material to kind of fill in all the spaces between these wires, in our case tubes or pipes. You want to fill in all that space so that you control nothing can go in unless it's going through that electrical cable, right? Or in our case, a pipe or a membrane tube. So that potting is kind of an important concept. You'll see it a lot, and that would be kind of adding stuff to fill that space, or to really control whether or not something can go through, right? So that would be it. That's really the one of the technical aspects. So the last form of membranes we see, and this is quite common, is, well, we basically take that tubular membrane system and we add lots more tubes and just make them smaller. Higher surface area, so long as we can keep them properly operating, so physically robust enough, not clogging by anything. We should have pretty good control over the quality of our water in terms of particles and stuff at this point. So as long as you keep good control of it, it should work very well, and you can just go as small as is practical, and then you have a whole lot more surface area for one cartridge than you could with either of the other ones. So hollow fiber membranes there are quite a common way to do it, and you can kind of see the expanded view here. And again, it would just be a matter of potting in between and around all of those tubes to make sure that no water can escape, and you can achieve your pressurization with that in place. Otherwise, it operates the exact same manner, and it's simply just higher surface area. So there's a few more physical constraints here, obviously. Things to worry about. Wouldn't want to crush any of them and prevent flow or something like that. But it is used quite often as a technology. Okay, so let's take a look at a problem. And this is a simple problem, or relatively simple. So essentially, it's going to be a mass balance, no reaction or anything. But it'll give you a good feel, I think, for how to approach a membrane type of problem. And I'll say this now. I would encourage you to draw the system of what you think it should look like, right, to begin. Okay, so it says we have here a membrane filter is used to concentrate an algae sample for culture analysis in a laboratory. So it's a little bit like our orange juice example, right? We want to concentrate it. That's actually our goal this time. So we have a membrane with a pore size of 0.45 micrometers is used, and it rejects 90% of the algae. The filtration cell has a volume of half a liter and can be operated in either cross-flow or dead-end mode. Initially, we're dealing with the dead-end mode. If the initial concentration of algae is 3.5 times 10 to the 5th cells per liter, and the initial volume is half a liter, it's 500 milliliters, what is the concentration of algae in the feed if 450 milliliters of solution is passed through the membrane? Okay, so what is the concentration left in the feed? So we'll say CF equals question mark after 450 milliliters of the 500 have been filtered.
then we're gonna run 90% of it through. And it rejects 90%. And that is not how, oh, it's not how it's supposed to be. Is permitted, what group gets retained is what's going to be called. We'll then just make it 0.5. What's our, oh, we have more stuff, of course. We have there. Well, I assume if it says the initial volume of the cell is 0, it means when we're pouring stuff in, it's filling up. There might be a different scenario for that. Is that along the right line of things?
Say it again.
If it says the initial volume, in this case, of like the second from the bottom line, the initial volume is 500, but earlier we have a filtration solvent volume of 5 milliliters. If it says the initial volume is like less than that, we just immediately assume it gets filled.
We have 5.
Like we have matching. We're wondering, we're curious if it's a redundancy or not. The initial volume being 500 milliliters and the existing volume of the filtration cell being 5 liters, 0.5 liters. Or is it like, there's going to be a scenario where the initial volume is 0 milliliters and they're going to have to fill up.
Okay, so did you draw it?
Not yet.
Okay.
I have
Draw a filtration cell that can contain 0.5 liters. Okay.
All right.
So draw that.
So it's because we're doing dead end mode. Yeah, it's the vertical one with something going in, something coming out, but we already know it's going out because once it comes straight to the in the feed, 450 milliliters of the solution is passed through the membrane. So it's 450 not necessarily, I'm looking there, it's 450 not necessarily the permeating.
Okay, so you draw a container that can
I'm with you there.
have a volume of 0.5 liters, right? Okay. Then you say the initial volume of the liquid is basically it's full, right? So you draw it, and so it is a little bit redundant, but you draw it, then you say, okay, here's the starting point, and then you pass most of it through, pass 450 milliliters of it through, and you're left with some remaining volume, right?
Right.
Some volume is remaining
So
here.
you pass 450 milliliters through. Wait. So not necessarily the feed is 450, not necessarily the permeate is 450.
The permeate is 450. Permeate, you permeated. You want to write it up that way.
Still trying to figure out what to do with the pore size. I mean, assuming that's like the areas.
Have I taught you anything to do with pore size yet? I mean, I told you about it, right?
Yeah.
But have I given you any equations, covered any of that?
I mean, I'm figuring that's like we could use that. That could be like the diameter of the pores. And we have to figure out
I could have also said it was a Shimadzu membrane, right? Maybe it's just a label. It's a red herring. It's not relevant. But this is why I was telling you it's simple, right?
If you have like
Straightforward.
2 red herrings per question, I can guarantee you my exam grades will drop tremendously. We just have things in there that are like, they gotta be useful for something. I can be sitting there for hours trying to solve it. Saber text knife.
But how do you deal with real life then? Some stuff doesn't matter.
Yeah, I can ISO it. I can be like, okay, this is gone.
Yeah, that's what I'm asking you to do.
I'm gonna freeze the layer. Freeze the core sizes. Okay. And we also have an initial concentration of that many cells per liter. We only have half of it. We only have half a liter. So then we literally just multiply 0.9 times our half. This seems pretty easy. Right? That would be how much gets retained. And then we have to figure out the concentration after that. I'm catching it. Okay. So then we're going to get that number of 1.9 because it retains 9% of the elements. Project. Yeah. So what's stuck in here, not in concentration mode, is that many cells about. Well, you also have to divide it by 2 because it's that many cells per liter, and we only have half a liter to go over. There's only half a liter in there. So then we're going to have to divide this by. This looks like a garbled mess.
Once I explain it, you'll be like, oh. I mean, and it sounds like you're getting to the right track. But.
Well, now I'm trying to quickly just now do like a concentration. Like we figured out roughly how many cells got retained because it's going to be whatever 0.9 and 0.9 times 0.5 times our initial concentration is what's retained on there. And then we just need to do that versus the volume. That's it.
Should be. I mean, if I understood you correctly. We'll go over it in a minute.
I don't like it, but that's what makes sense to me. Yeah, we divided by 2, multiplied by 2. Yeah. I didn't realize that you needed to. Yeah, just let those cancel out. I just. Yeah. But I guess like if I'm having to collaborate this much on one question, I can only imagine this will work out.
Stop. It won't be that bad.
To get the brain going. Yeah, that's a good way to express it.
All right, so I'll go ahead and go through the problem with you. There's probably a couple ways you can do this. I think some of you may have been overthinking it, but a little bit. So we did talk about, let's say, pore size, but I didn't really give you anything to do with it, right? We didn't talk about, aside from generically smaller pore sizes, more pressure. I didn't even tell you what we do with pressure yet, right? So we'll cover those topics and like some equations for them next time. So for this one, as I said earlier, it's a lot like when we have a pipe and it splits into 2. We expect no concentration difference normally, but with a membrane, we can achieve that, and so it's a mass balance. There's no reactions going on, but we have a mass balance here of the same amount of stuff. Is it like we start with this some amount of stuff, some amount of cells in this case, but we're going to end with the same amount, right? So we have a conservation of mass or of number and of our volume, right? So in that sense, if I just say that, that's approximately enough to get you through the problem. Right? We're not actually applying too much of the membrane stuff. This is just in the context of membranes, a mass balance happening. So I think the way that might make the most sense is to take a look at the number of cells. We were actually given an initial concentration of cells. So we were given, I guess, a C, I'll say a C initial, C naught. That's like the C feed before we push any water through. And so the CF, I guess, I'll say final. C feed final, maybe. Not quite sure what the best way to write that would be, but like that's what we're looking for. But we were given some C initial that was 3.5 times 10 to the fifth cells per liter. So the number of cells we have in our half a liter to start with, we can just track the number of cells and say, okay, we have this number to start with. Some of them escaped through the membrane, but most of them are still in there, and we've reduced the amount of water. So we're just going to basically just change the denominator. If we could even start and say, well, what if 100% was, if it was 100%, we could just do a change of volume and that's it, right? And in fact, our answer here with a 90% should be about 10% different from if we just assumed it removed all cells, right? If this was 0%, there'd be no difference, right? The concentration wouldn't change. The cells are going through, right? So that would be quite easy. But maybe a good way to look at it would be to say, okay, the number of cells we start with are 3.5 times 10 to the fifth cells per liter, but we have to multiply that by half a liter. And that's going to give us, 1.75 cells in our solution. Okay, so then we can count how many escape and subtract that, so we can say cells, so number of cells, what I'll do is I'll say number of cells escape. And I'm going to subtract that from the number of cells we started with. So we'll say number of cells start minus the number of cells escape, divide that by the new volume we have, should give us our C final. So that would be one way to do it, right? You could also just keep it in concentrations and find it that way as well. That'll be our C final, our C in the feed at the final condition, we'll say. Okay, so that means we've got, sorry, I said 1.75. 1.75 times 10 to the fifth cells. So we started with that many. We're going to multiply that number of cells by 450 milliliters, or 0.45 liters, because that's the amount of water that went through. So we'll take that and say, well, first of all, if we're trying to find the amount escaped, actually, let's do it this way. Let's say the number escaped and solve for it. So number cells escape should be equal to, in that volume, we have some concentration and some percentage that escape, right? So what we're going to say is, we'll take, or I guess concentration, we should use number, right? So we'll take that volume, no, the concentration was correct. So we'll take the volume, 0.45 liters, multiply that by our number of cells per liter, which would be the 3.5 times 10 to the fifth cells per liter, and then say, okay, 10% of those escaped out, because I'm counting how many escaped right now. So I'll say that's multiplied by 0.1. So we have a number escaping, and we can do that math real quick. That's going to be 3.5 times 10 to the fourth times 0.45. And if you look here, what I've done is even though I calculated the actual raw number a minute ago, I realized it's going to be easier to stick with the concentration here. And I just made sure to cancel the liters here and the liters here. So, I had to revise my approach a tiny bit. So I'm just trying to explain that out loud here. So we can put that in a calculator, and essentially that's going to be here. I can just take that 0.1 and then reduce the times 10 to the fifth down to times 10 to the fourth for that. But I can take 0.45 times 3.5. So it'll be 1.575 times 10 to the fourth. So that's the number that escaped. We could probably go based on how many were retained, by the way, and just work the algebra a little different, do the mass balance a little different. We could probably do it that way too. But since this is the way I'm going, that means that this C final in the feed is going to be equal to 1.75 times 10 to the fifth minus 1.575 times 10 to the fourth divided by our new volume, 0.05 liters. Okay, and it's probably a lot to do in just the calculator. I'll pull up. So, does that make sense? We have done that, and you can see that if we had complete rejection, we don't have to subtract any that escaped. We would just keep that starting amount and divide it by the new volume, right? And so in a way, it's just like concentration times volume over and over again, right? So we said the number escape was 1.575 times 10 to the fourth cells. The number initial, that was 1.75 times 10 to the fifth cells. So then the C final in the feed would be the initial minus that final, and divide all of that by that new volume, which was 50 milliliters or 0.05 liters. So our final concentration is going to be in cells per liter then at 3.19 times 10 to the 6. Did I actually show you guys that? Or were you looking at Excel? Okay. I got confused there for a second because I was like, oh, I need to move this out of my way. Did you even see it? Sorry. Okay, good. So that was so that would be our answer then. Does that make sense? You see now how it was like really was sort of just mass balance. Filtration stuff was just added context. So we'll get in next time to more of the how does the pressure affect the permeate, and how's that fouling resistance work, some things like that to give us some more equations. But I wanted to start here because I want to make sure that you're recognizing that the basis of this is just kind of the same mass, the principles of conservation of mass or number that we've been dealing with, right? So try not to overthink it. It's pretty common to see somebody describe what kind of membrane they use in terms of the pore size. That doesn't usually affect our calculations because usually there's other parameters like permeability or porosity or membrane resistance that we're actually going to use. So usually something like a pore size description is not actually relevant to our calculations. Just a side note there. Okay, so we'll see you on Tuesday next week. I do have some exams. If you didn't get yours already, feel free to come up and have a good weekend.